This is our website on reproduction. We believe in the right environment we can hatch a chicken egg without the use of a shell. However, some people don't think this can be done because the egg gets all the nutrients from the shell. But we discovered that we can make an artificial shell that does the same thing the actual shell does. Next, we have our hypothesis. If we create a shellless culture, then we can incubate a baby chick without the use of a shell. We test our hypothesis by making a shellless culture by putting water into the base of a plastic cup, then making a sling with saran wrap that held calcium carbonate, water, and the chicken egg. Then we put it into the incubator. We accept our hypothesis because we know it can be done. However, when we did it, we had some human errors causing us not to be able to be successful. The materials we used were a plastic cup, cling film, metal rod, coffee cup lid, calcium carbonate, distilled water, benzalkonium chloride 0.1%, eggs, round bottom glass, rubber gloves, disinfected, incubator, dropper, and Lysol wipes. Next, we have some background information. Some of our background information was only broken into two different groups in our class. One group had to find out how to build an incubator, and one group had to find out how we could get the calcium. The group that decided to find the calcium, we put water in the base of a plastic cup, then made a sling with saran wrap that held calcium carbonate, water, and the chicken egg. The other group had to build an incubator. First, they had to see if they could build an incubator from home. After they found out they could, they started looking for steps to show them how to build one. Eventually, they found a way to build one, but they made some of it up as if they found a better way to build it. Now we have the shellless culture. We had three questions. Can you hatch a chicken egg without a shell and have it survive in a natural climate? 2. Is it possible to build your own incubator that is capable of hatching a chicken egg? And 3. Would the life cycle of a chicken hatched without the egg have the same life cycle as a chicken hatching its own egg? Can a chicken be hatched without a shell? The answer is yes. If you make the right environment with the right climate, the chicken egg will survive. Also, if you put the chicken in its natural environment, it would survive as long as you put it in its normal environment after it strengthens up a bit. The life cycle of the chicken egg would not be the same when it is a chick because even though it is getting the right nutrients, it still wouldn't work as well as the original egg. However, when the chick grows up and gets stronger, it should be having the normal life cycle just like any other chicken. For the artificial shell, it needs to be secured to keep out bacteria. However, it needs to be semi-permeable to make sure oxygen and carbon dioxide can get in and out along with essential nutrients. Since the shell needs to be fully secured, we would use a plastic cup. We would put the egg in the plastic cup, then seal it with saran wrap. Most of the nutrients it needs would come from the yolk, but you also need the calcium, so you can use calcium carbonate to give it calcium. Next, we have the were we successful? We were not successful. Our pre-incubation started November 25th, 2016, and we ended the incubation on December 5th, 2016. At the start of the incubation, it looked like some chicks were forming, but by around the 10th day, it appeared that everything stopped forming, so we stopped incubating. In addition, there were multiple reasons why we weren't successful. For example, we had some human errors like accidentally dropping or cracking the eggs, but we also had some eggs that weren't viable. Those are the main reasons why we weren't successful with getting the chicks to be produced. We had one chick alive, however the organs stopped forming. Next, we have some of our data. We have quantitative and qualitative data. Our quantitative data is that we had 8 eggs to start. One got dropped, one got cracked, and not all the eggs were viable. All the eggs that weren't cracked remained in the incubator for about 10 days. However, there was very little to no progress, so they came out of the incubator. The temperature ended up varying because the incubator couldn't keep the eggs in the right temperature. In the base of the cup, we put 10 drops of water, and in the sling of the cup, we put a tiny bit of calcium carbonate, a little bit of water, then the egg. Qualitative data. When the eggs when the eggs were in the incubator, it looked like some organs in the heart started to form in one of the eggs. However, we waited a couple more days and nothing happened, so we made the decision as a class to remove all the eggs from the incubator because they weren't viable. For incubating, we made a shellless culture with cups, so we put water into the base of the cups, then we made a sling with saran wrap. In the sling, we put a little calcium carbonate and water in the sling. Finally, we put the egg on top of the sling. DNA and genetic traits. Every living cell has DNA. DNA is located in the nucleus and makes up everything special about you. Also, DNA can self-replicate. DNA makes up your genes, which also makes every individual special in some way. Genetic means of or relating to genes or heredity, so genetic traits are the different traits people get from their parents. Asexual reproduction is a type of reproduction that produces offspring from a single organism. The offspring gets the traits of that parent only, and it does not involve with the fusion of gametes. Finally, the offspring will never change their amount of chromosomes. Sexual reproduction is the production of new living organisms by combining genetic information from two individuals of different types. A gamete is a mature haploid male or female germ cell that is able to unite with another of the opposite sex in sexual reproduction to form a zygote. A diploid cell resulting from the fusion of two haploid gametes, a fertilized ovum. Clone. An organism or cell or a group of organisms or cells produced sexually from one ancestor stock to which they are genetically identical. Mutation is the changing structure of a gene. Next we have meiosis. Meiosis is a type of cell division that reduces the number of chromosomes in the parent cell by half and produces 4 gamete cells. This process is required to produce egg and sperm cells for sexual reproduction. There are 9 stages of meiosis. They are divided into stages when the cell separates the first time and then the second. Now we have mitosis. Mitosis is the process where a single cell divides into 2 identical daughter cells. During mitosis, one cell divides in once to form 2 identical cells. The major purpose of mitosis is for growth and to replace worn-out cell. Each daughter cell has the same genetic code and the same number of chromosomes. Mitosis is divided into 5 stages: interphase, prophase, metaphase, anaphase, and telophase. Finally, we have our conclusion. In conclusion, our project was about seeing if we could incubate a chick without a shell. Also, on this website, we talked about DNA, genetic traits, mitosis, meiosis, and if we were successful in incubating a chick. Furthermore, our hypothesis is if we create a shellless culture, then we can incubate a baby chick without the use of a shell. Also, we accepted our hypothesis because we have seen it be done before. However, a couple human errors kept us from being successful. In the end, the results were not what we were looking for, but we can learn from our mistakes for purposes later on. We also have our bibliography. We use 6 different websites. Thanks for listening.